I'd like to do a quick review of ChiSquared tests. ChiSquared tests are simple tests that we use throughout genetics, particularly you'll use them in the first half of this course a lot, to look at some very simple questions. And I know each of you have probably done ChiSquared tests before, but I think it's useful in genetics to review the model, talk a little bit about the way that the calculations are done, and also explain how we're going to do it in genetics. Now, chi-squared tests are, as I said, are simple statistical tests asking a very straightforward question. You have these observed counts, and we will usually do that in the form of progeny or something, and say we observed this many progeny of this type. And the question becomes, is that what we expected to see? Did we expect to see 5 blue progeny to 7 red ones or whatever? Now, in genetics, our expected counts over here, they will come from the crosses that we make and then the resulting expected ratios. And we're simply asking, do the data that we see fit the ratios that we would predict them to? The calculation itself is very simple. It's just observed counts minus expected squared divided by expected. If there is a perfect fit, if we get exactly what we expected in our observed, then this will all, of course, be 0. If it's not that, and it usually isn't, we have some number greater than that, and that becomes what's interesting. The question becomes, do we depart from this perfect expectation? Is that significant enough or important enough to be a significant departure, or is it just a chance departure? Would you often expect to see that much departure just based on natural process? And that's really what a chi-squared test does. Now, mechanically, a chi-squared test, we would do this for each category. If we had 3 categories of progeny, red, white, and blue, we do it for red, we do it for blue, whatever, then we sum it up. This is a summation signal over here. We would take the sum of all those, and when we did, we would get a chi-squared observed value. And this is the actual value that we would use to determine whether the overall fit to the ratio is what we expect or not. Now, in genetics, what we've done is that we put this into table form, so that we have a phenotype or genotypes over here, just a category, red and white or blue and green, whatever it might be. Your observed data here, your expected data or expected outcomes determined from taking these ratios. We'll talk more about that in a moment. Observed minus expected, found here, which is the top part of this equation. Observed minus expected squared, so we just take this cell and we square it. And then the observed minus the expected squared divided by the expected. That's this whole part of the equation right here. Once you total up that column, you have the total observed chi-squared value, which is equal to this chi-squared notation here. With that, plus the degrees of freedom are determined by the number of categories you have minus 1, n minus 1. So in this case, you might have 2 categories, red and blue. That would be 2 minus 1, be 1 degree of freedom. These are then used to develop what is known as the table value to figure out what value you'll be comparing to. You make a comparison between the table value and the observed value, and you determine whether you can reject or fail to reject this ratio that you're interested in. Well, let's try one of these. I think it'll make more sense if we just work through one very quickly. So here's one from your lab manual, page 116. In this particular case, out of 225 students, 141 are identified as female, and 84 identify themselves as males. Now, with humans, human primary sex ratio is approximately a 1-to-1. So in classes like genetics, we would on average expect about a 50-50 ratio, about half of them to be male, about half to be female. Obviously, this isn't the case here. We've got more females than males. If it had been a 1-to-1, we'd have exactly equal numbers or approximately equal numbers of the 2 genders, but we don't have that. The question becomes, is this a significant departure? Is this something, a large departure from a 1-to-1? Or is this something that just might occur anytime we take a draw of 225 individuals? In other words, is it common to see this much of a difference? Well, to do that, we're going to have to do the chi-squared analysis, and we will have to determine the degrees of freedom. We will determine our observed chi-square statistic, and then we'll use those to determine a probability level for this observation. Now let's go to our table. I've already filled in that we've got males and females, our 2 genders here. Our observed data, of course, is going to be 141 and 84 as of 225. Now we got to get our expected, because we need observed minus expected in order to do this. Well, our expected comes from this ratio of 1-to-1 that's right here. Okay, we have 1 plus 1 is 2, So 1/2 are expected to be males and 1/2 are expected to be females. This could have been any ratio. It could have been 3 to 1 or some 9 to 7. You simply add them up. If it's 3 to 1, you'd have 4 parts. So 3/4 would be one gender and 1/4 would be the other. That won't make any sense with gender, but it'll certainly make sense with other traits as we go through. But in our case, we expect 1/2 and 1/2 to be each. So 1/2 of our total tells us how many we expect. 1/2 of 225 is 112.5. Don't worry about the fact that you got half an individual here, but that's what we expect to see. Or if you're doing it on a calculator, of course, it's in the decimal form of 0.5. Now, these are going to be our expected. They're going to be the same expected for both the males and the females because we're looking at a 1-to-1 ratio. So if we fill those in, we have this total right here. Now notice that it's very important that you just quickly check to make sure that your expecteds total up to the same as your observed. Now within rounding error. This will be very important, a little quick check, because if you get to some more complicated ratios later on, you may have made a mistake somewhere. And if these don't add up to the same number, then you know you've made a mistake, and there's no reason to go through the rest of the calculations because they'll be wrong. So it's a quick, easy way to make your first check. Now we've got observed, we've got our expected, so we can start filling in the lines across here. Observed minus expected is 141 minus 112.5 is apparently 28.5. 84 minus 112.5 is apparently minus 28.5. Once we square those, the negatives go away. That gives us 812.25. Okay. Now we have all the parts we need. We have our observed, we have our expected, we've squared those differences. Now we can complete the last part. We can take our observed, which has been squared, our observed minus expected, which has been squared, is 812.25, divided by our expected, and that will give us a value for each of these cells. Okay? Now just to show you in the regular equation, there's our observed, our expected, we squared it, divided by the expected, and for each gender, for females we get 7.22. In this particular case, because we're using the same numbers, it's going to be the same for males. If we were using a 3-to-1 ratio, this might be a large number down here for females and be a small number for males or something, whatever. We just bring whatever expected we have for that particular phenotype over, okay? So if we do this, we got 7.22, 7.22 adds up to 14.44. Observed chi-square value then is 14.44. Our degrees of freedom are 1, as we talked about before, because we have two groups, males and females. So n is 2, n minus 1, or 2 minus 1 is 1. Now we need our table value, and then we'll use that to reach our overall conclusion down here. Okay, here's the table that's found in your lab manual, and it's a typical chi-squared table. There are two things you need. One is a column with degrees of freedom, which we've already determined to be 1, so we'll put it right there. And then we need a column of probabilities over here. Now there are multiple ways to use this table. We're going to use it in a simple way. We're going to just take values from one column. The one column has been sort of highlighted here in light blue. We're going to use 0.05. It's a standard value to use by most biologists in most situations. It means that 95% is what we're interested in. 95% of the values fall on this side of the number, 5% fall on that side of the number. Okay? All right, the way this works is that we simply take the degrees of freedom, follow it over, we go down this column, and where they intersect, that is our table value, as we're going to call it. Our chi-squared table value, our test value, is 3.84. Okay? So we're going to look for numbers either larger or smaller than that and use it as an absolute. Now what this really means is that if you look at this distribution of chi-squared, 3.84 falls right here, and 95% of the numbers that you would get by doing a chi-squared calculation should fall below that number, just by chance. About 5% will fall above that number in this red part of the tail. Now if we wanted to be more precise about it, we could go to 99%, okay, or 0.01. That would be a smaller tail out here somewhere. Or we can even go to 99.9%, which would make a very small tail. Okay, but we're going to go with 95%. So 95% of our numbers should fall down here. We'll just call those the chance deviation numbers from 0. 5% will fall above that, and those in the red are those that we'll determine to be significant. So essentially, if you're on this side of the number, you're not significant. In other words, you're not going to say that you deviate from the expected ratio. If you're on this side, larger, then you're going to say that you do in fact deviate from your expected ratio. Well, now that we have our table value, let's go back and put it in play. We have our table value. We now can look and basically directly compare our observed chi-square versus our table value. Our observed chi-square of 14.44 is much larger, much larger than 3.84, meaning that it's way out in the distribution out here somewhere. It's not a number that you would commonly see if you actually had a 1-to-1 ratio occurring. In other words, this is not a common amount of male to females if in fact it truly was a 1-to-1 relationship underneath there. Larger than our table number. It's way out on the end of the distribution. We're going to reject the idea that these data fall under a 1-to-1 ratio. Okay? We simply reject that idea. And we can see there's a lot more females than males, and we significantly say that there are significantly more females than males in this particular class. Now that could be interesting and important. It could be, why are there more females here? Why do more females take genetics or whatever? Or it could be a case of where we just need to go and take genetics classes from say 4, 5, or 6 semesters, total all those up so we have several thousand students, and you might in fact come much closer to a 1-to-1, which in fact is probably what will happen. The ratio in genetics is usually about 1-to-1. Okay, but in our case, we've found a ratio. We've tested that ratio against our observed data, and we say that it does not fit that ratio. We reject the hypothesis that we have a perfect 1-to-1 fit or even a close 1-to-1 fit. Okay? Now that's a simple chi-square with just 2. What I'd like to do is go ahead, we won't be using it in this week's lab, first week's lab, but you will be using it throughout the rest of the labs that would deal with the flies and fly colony. So let's go and do a slightly larger chi-squared ratio. In this case, all we're going to change is that we've got more categories. Okay? Instead of having just male and female or red and blue, we now have 4 categories. And we have observed data. We have 178 red, 57 blue, 193 white, 64 or 62 greens. Now I've already put them down here in my observed column. The question is, what about my expected? Well, I need some ratio. And we could use any ratio, but we'll say that this came from a cross that generated a 3-1-3-1 expected ratio. Okay. Now, what you can do is you can match up the 3s to the largest and the 1s to the smallest, but based on crosses that you'll do, there'll be a fixed expectation. You'll expect so many reds to so many blues to so many whites. We'll say that that expectation here is that we would want 3 reds to every 1 blue to every 3 whites to every 1 green. Okay. Now, how do we get our actual expected counts? Well, that's done by taking these and saying 3 plus 1 plus 3 plus 1 is 8. So 3/8 should be red. 3/8 of whatever this totals up to should be red. 1/8 should be blue, etc. So we simply multiply those out. We say 3/8 should be one group, the other. That will give our expected. In doing so, we see that we expect 180 to be red, 60 to be blue, 180 white, 60 green. Again, we check to make sure that totals up to 480, which is the same as our observed, so we feel fairly confident that we're doing okay here. Now we have a simple pass-through. We have our observed minus our expected. We can just do the mathematics of that. Okay, we can take 170 minus 180 is minus 10, square that's 100, square 4, and we get 16. Do our observed squared minus divided by expected, and we get these small values that total up to a total of 1.829 or 1.83. If you want to round up, that's fine. Okay, so now is this significant or not? That becomes a question. Is our observed value of 1.83 significantly different from 0? In other words, do these data depart from a 3-1-3-1 ratio? That's what we're asking. Remember, this would be perfectly 0 if it was absolutely observed exactly what we expected. But clearly we didn't. Here's 193 and we only expected 180. So is that significantly different from 0? Well, what we have to do is of course have our degrees of freedom. In this case, we've got 1, 2, 3, 4 categories. Because we have 4 categories, n is 4, so n minus 1 is 3, so we have 3 degrees of freedom. All right. So now we can go back to our table. We're going to again use the same column, 0.05, but this time we're going to move down and we're going to use 3 degrees of freedom. So we're just going to cross at a different area. And when we do, we see that now our table value has gone up because we have more degrees of freedom, more possibility of a chance event. Okay? And now our table value is 7.82. So the question is, given our observed value, which is 1.83, is it larger or smaller than 7.82? Well, obviously it's much, much larger than 7.82, our table value. So we are on this side of the answer, and when we're on this side of the table, that means we fail to reject. We say that there's no evidence from the data we've collected that we violate a 3:1:3:1 ratio. Now, it doesn't mean the 3:1:3:1 is the correct one. It just means that we do not in our data have the ability to reject that the underlying data actually came from that particular ratio. Okay? You'll get a chance to try all sorts of ratios, 3:1:3:1, 1:1:1:1, 9:3:3:1, etc. And also shouldn't point out that chi-squared can be used as a goodness of fit test. In some cases later in the semester, you'll be asked to decide between a 1:1 ratio and say a 9:7 ratio. Which one of those fits the data the best? Simply do two chi-squared, one with a 1:1 and one with a 9:7, and see which one gives us the smallest observed, because that will be the one that has the best fit. Again, remember, because we're trying to go towards 0. So you'll use chi-squares in all sorts of different ways.